query I have is those benign bronze plaques are, if you don't.
Also, it's we come to the next lecture, and you have already experienced that. I mean, it's very healthy what we're doing. It's like sauna. I mean, we have one temperature, and then we go into the cold bath. Prayag, I don't know whether you are the cold bath, but nonetheless, it's a shocking change of temperature and topic. We have talked about the axolotl before, and I think this is what we'll hear about, very molecular research. Prayag comes from India. He trained in India, and then he came to Germany, first working at and studying at the Max Planck Institute in Dresden, and then he worked on axolotl, and he went with the most famous laboratory in the world with Ili Tanaka to Vienna at also very famous institution, the Institute of Molecular Pharmacology there. And then we were able to recruit him 3 years ago, almost now, 2 years, to MDIBL. And we will have, after lunch or between our lectures and lunch, a short visit to his animals, because he is now the king of how many axolotls down there?
1,000.
Okay, so you will see, have a quick view of his kingdom. And now he's going to explain to us the powers, the mystical powers, but you are actually unraveling everything of the axolotl.
Thank you, Herman, for a very kind introduction. My lab essentially works with axolotl, but we are also interested in methodology. But today, let's start with, how do I change it? Wait. Yeah, okay. So yesterday I was seeing Daphne, and then I was wondering what is the closest in medical terminology a Daphne could be. And it might shock all of you, but there is a disease which is called tree syndrome. In this disease, essentially the hand grows out as you can see, there is a lot of malformation at the tip of the hand, and it looks like a tree. So I was actually wondering if that's what was Daphne. Maybe not, I hope not, but something to ponder upon. So I'll stop my art lessons here and move to the science, which I'm more comfortable with. So why do we need organs? It's because as we age, we suffer injuries and we require replacements. And as you know, the most common way of doing that is, wait, which one is the, oops, I escaped. That's the mistake I did, like everyone. So which one is it, the bottom one or the center one? Oh, it's at the back. I see. Oh, okay, good. Okay, so as you know, when you lose a limb, today we use prosthetics to replace the limb. But the wonder of science is if we can regrow some of the structures on site. And this is what we are learning in our laboratory. And to do that, we use number of model organisms. Here I'm showing you, this is not what we work with, this is a planaria, but this is one of the extreme of tissue regeneration. This animal, essentially you cut it at any plane. So like here, it is cut into 3 parts, and all 3 will become 3 different organisms. So this is a full body regeneration that we are talking here. You can also cut this animal vertically, little bit on the side, diagonally, triangle, rectangle, whatever shape you like, and it will make a new creature essentially out of all these pieces that you see here. So, but tissue regeneration, when you look at in the animal kingdom, it's not something new. It has been existing across the animal kingdom. So, for example, Hydra, you can cut it, and there is also a mythological figure, Hydra, as you know. It will make new head. Similarly, planaria we discussed. Earthworm, you can cut it into two. Starfish, they can lose their tentacles and they will regrow that. Cockroach can also lose their legs and they will regrow it. Zebrafish are known to regenerate their fins. Xenopus can also regenerate, and lizard tail already we heard yesterday. But among all, my lab is interested in this wonderful creature called axolotl, or ajolotl as it's pronounced in Spanish. This animal comes from Mexico, and it's a champion of vertebrate regeneration because it can regenerate many different body parts. You can cut their legs, tail, damage their heart, lung, liver, and even brain, and it will regrow all those structures. So what we are interested is, can we learn something from this animal? And then if we know how it functions, then we can look at mammalian models, which is the mouse and human, and understand why we cannot regenerate. So I won't go into the detail, but in the past, we have published this paper where we have shown that when the axolotl limb regeneration happens, it happens via de-differentiation of fibroblasts. So just like human, they have fibroblast in their hands and arms, and this fibroblast magically, not magically, but basically there are signaling cascade that kind of converts this fibroblast into embryonic-like progenitor cells, and these progenitors then go on and give rise to a full limb. But I promised Herman that I won't talk about limb. Today is tail and metamorphosis. So let's switch gears to the tail. So apart from limb, we also study tail. Why we study tail is because it's a primary body axis. So when scientifically we look at our body, this is the primary body axis, and hands and legs are secondary body axis. And there are very, very few animals on the planet that can actually do primary body regeneration, and this is one of them. So you amputate, it will form a blastema, and then it will go on and recapitulate and make a new tail. The reason, as I mentioned, it's a primary body axis, and when it regenerates, it restores all this pattern that you see. For example, these are the muscle bundles, these are the vertebrae. So this is of immense interest. Like, when you suffer an injury, and if your vertebrae are damaged, your spinal cord is damaged, can you repair it? So the question we asked is, when the blastema formation happens, what kind of cells move in to form the blastema, and what kind of genetic and molecular changes happens in those cells so that it can form the blastema? So these are the questions. So before I go into that, the regeneration in this animal is fully functional. I just want to highlight once again. And you can see that on your left, you have an uninjured animal. On the right-hand side, you have an injured animal which has regenerated. And both these animals can show this kind of a C-curve movement. That's what we call. So they are fully capable of recovering from the injury. Now, I'm going to go a little bit into the developmental biology, which is the how essentially our primary body axis develops. So there are two kinds of animals. You have vertebrates and invertebrates. Vertebrates have the vertebrae column that we have the spinal cord vertebrae. And then there are animals like planaria that does not have those vertebrae. So the formation of vertebrae essentially begins with this particular process called somatogenesis. There's a video, but okay. So it begins with the process of somatogenesis, where you have this structure called presomatic mesoderm, which then segments and forms this kind of balls, which are called somite balls. These are somites. And each of these somite will give rise to one vertebrae in the body. So axolotl being a vertebrae, basically, it's no different from mouse or human. It has these somites, and it gives rise to the vertebrae. That's the point of this slide. Now, what is the textbook knowledge? The textbook suggests that you have a neural tube. This is your, eventually this is gonna become your spinal cord. You have a notochord, and you have a somite. And this somite, there are cells in the somite, as you see here, and the cells from this ventral part of the somite starts to kind of move out, delaminate and move out, and then they cover the notochord, And that's what becomes eventually vertebrae. Whereas the dorsal part of the somite becomes dermatome and myotome. Dermatome give rise to the fibroblast in your primary body axis, and myotome give rise to the myogenic lineage, which is your muscles in your primary body axis. The embryonic development pathways have been worked out very nicely. People understand how the differentiation happens. Like you have the presomatic mesoderm form the somite ball, and then somite goes into dermatome, myotome, and sclerotome. Sclerotome is your vertebrae. These three distinct lineages. And as they transition through this various steps, there is one particular marker that I want to highlight. There is a gene called MEOX, and this gene is present in presomatic mesoderm, then it's there in the somatic progenitor, and then MEOX is lost so that it can give rise to your dermatome, fibroblast, myotome, which is your muscles, and sclerotome, that's your vertebrae. Okay, so from the introduction, I want you to remember three things, and that's somatogenesis lead to the formation of somites. Somite contains the somatic progenitors, and the somatic progenitors can be identified by an expression of one particular gene that's called MEOX, and then the somatic progenitors differentiate into various mesodermal lineages. That's your muscles, fibroblast, and skeleton in your primary body axis. So when we started working, we were interested in this somatic lineages. So the somatic mesodermal lineage that is highlighted here by muscle fibers, these are your myomeres that are situated there. Then there are fibroblasts that sits inside this muscle or in the gaps of this muscle. So these are called interstitial fibroblast-like cells. And you have some other fibroblasts that sit in the fin, which is called fin mesenchyme. And the question we asked is, how does each of this cell type kind of participate during tail regeneration? Now, logically, when we think about the process of blastema formation and cellular participation, there are two things that come to our mind. The first is there are pre-existing stem cells or progenitor cells, and these progenitor cells just proliferate and differentiate to give rise to different cell types. The second way is you have a differentiated cell, like we have shown in that earlier paper of limb. You have a fibroblast, and this fibroblast kind of de-differentiate and go back to your embryonic identity. That's how they become progenitor. And then this progenitor give rise to various lineages. So we were interested in this, and we wanted to challenge our hypothesis, which one of that is true. So we used a technology called inducible Kree-Lox-P. I won't go into the detail of this because of the nature of this conference, but what this technology allows us is to label individual cell type permanently. So you can label a certain cell, and then you can track those cells as the regeneration is happening. And when that happens, the question is whether your cell remains in the same form, so first question is whether it participates or not. And second question, whether it remains in the same form or it becomes something else. And you can track it. So using this technology, we made some transgenic axolotls, which allow us to label certain cell type as seen in this picture. And then you can see that there are no muscles labeled in this tail. But when we make an amputation, cut the tail and allow it to regenerate, then suddenly you start seeing this red color cells, which appear to be muscles. So this is nicely visible here on this pseudo coloring that we have done. We have a black and a gold here. So you can see that before amputation, our transgenic line labels three distinct populations. We have a vertebrae, we have intermyotomal fibroblast, and we have a fin mesenchyme. But it does not label myotome or a muscle lineage. But when we amputate and allow it to regenerate, then suddenly this muscle-like structures appear. So this is very interesting. And we, as a scientist, we have to answer certain questions like, how clean is our line? Ondin is sitting there, she would probably ask me this question. So we have to prove that our line is clean and it's not. So muscles traditionally are known to form from a muscle progenitors, which can be identified via a certain expression of a certain gene that's called PAX7. So the PAX7 here is labeled in green, and our cells, these cherry cells are labeled in red. So our cells are distinct from this PAX7 progenitors, which are the known progenitors of the muscle lineage. So, and then when you allow it to regenerate, we see this, that some cherry positive cells turn on PAX7, for example, in this yellow arrowhead. So initially you have a cherry cell, but then it has a capability to turn on PAX7 progenitor cell, and then they go on and make muscle fibers. So what we have uncovered here is that we have a cell type that is one lineage up of the PAX7 progenitor. Now, this is, although I showed you that it happens during regeneration, this phenomenon we also uncovered during wound injury. So if you damage the muscle and then you allow it to heal, you see that it's participating. If we label this transgenic line early enough during embryonic development, then we also see that some of these cells have the capability to participate even without injury. So this gave us the very first hint about these cells that we are talking about. These cells that are capable of giving rise to muscle lineage exist during embryonic development and can participate during wound healing and regeneration. So to further understand, we developed another transgenic line, and this transgenic line labels the fin mesenchyme and vertebrae. But as you can notice here, there is a complete absence of this intermyotomal fibroblast. And now, when we amputate and allow it to regenerate, we notice a negative selection, which means this unlabeled population in the intermyotomal space is giving rise to most of the tail. This indicates, basically, these results indicate that our cells reside in this intermyotomal space between the muscles. Now, we combined it with one of the latest methodology in the field, which is called single cell sequencing. So what we do here is we take a tail, we dissociate all the cells, and then we ask how many different kind of cells reside in the tail. So the way to do it is so before this technology, a scientist would section the tail, and based on the morphology of the cell, they would identify a cell. But what this technology allows us is that we look at the transcriptional signature. Transcriptional signature is what kind of genes are expressed in individual cells. And if the genes expressed in two different cells are similar, then we call that, okay, these are the same group of cells. So that's what we did. And so you can sequence thousands and thousands of cells and look at what kind of genes are expressed in these cells. And then you can group them and say, okay, this group of cells are similar, that group of cells are similar, this group of cells are similar. So, and then you can look at what are the genes expressed in them. So we took this animal, we separated these cherry positive cells from the non-cherry cells, these red color cells, and then we performed the single cell sequencing and looking at what are the genes expressed in them. And what we found is that this is just after conversion. At this time, there is no muscle contribution from this transgenic animal. And what we found is that we could identify the fin mesenchyme cells, which is identified by certain genes. This is TWIS3 and PRX1. We could also identify skeletogenic lineage, which is your cartilage vertebrae. That can be identified by expression of PKD-CC and CHRDL1. These are the name of some genes, essentially, that kind of impart the identity of skeleton. But interestingly, we also identified a group of cells that expresses these two genes, sclerexis and meox. And as I mentioned earlier, meox is a marker of progenitor state. So now this is one single cell sequencing data I'm showing you, but we have done many, many more single cell sequencing from uninjured tail and during regeneration. And we found that there are these progenitors, and these progenitors have the capability to undergo this kind of a lineage commitment. They kind of give rise to myogenic lineage, skeletogenic lineage, and fibroblastic lineage this way. So this is the first data in the axolotl that kind of shows that unlike humans who have lost this meox identity and cells, we have these progenitors in axolotl. They persist throughout their life, and then they are capable of giving rise to all these cell types that I was showing you. So here is a plot. It looks like a mess, but the more darker green it is, it means you have an expression of a gene. That's what it depicts. And you can see that this group of cells, which I'm calling progenitor, have an expression of meox, sclerexis, lunatic fringe, and tenomodulin. These are the markers of the progenitors. And by these markers, we can identify that these are indeed those progenitors sitting in the intermyotomal space. Here are markers for fin mesenchyme. So you can see that there is more green signature here, and then there is more green signature here in the PR exon. So that labels the dermatomic lineage. The CHRDL1, SOX9, and OTOS labels the committed chondrocytic lineage. So you can see that they are more expressed in this group of cell. So I forgot to mention here, but every dot here is a cell, and the color indicates how much of that gene is expressed inside those cells. So and then on the top right, you can see the myogenic lineage. So PAC7 and MIP5, these are the markers. These are the genes that are expressed in muscle lineage. So we could see very nicely how these progenitors are kind of, differentiating. They're giving rise to different cellular lineages. Now then we took it further. Now that we know what are the genes for the progenitors, can we really see them in the tail on the side? And for that, we used another methodology, which is called HCR in C2 hybridization, which essentially allows us to identify which cells are expressing these genes. And you can see here that between this muscle bundle, so this green thing that you see here is the muscle bundle, and in between those muscle bundle, the cells are nicely lined up, and they are expressing all these genes, lunatic fringe, meox, and sclerexis. So one more in situ image, basically, this is during tail regeneration, and we find that this meox positive cells are nicely sitting here and here between this. So this is the muscle, and then as the tail is regenerating, they kind of pop up again and again, and that's how the tail regeneration happens. We have also done the staining for a proliferation marker, and we find that these cells that are sitting in this intermyotomal space they show the maximum proliferation. So what we have uncovered here is that before amputation, there are two kinds of cells. We have a meox positive cells and PAC7 cells, and both of them then move on into the blastema, and then both the cell population kind of contributes to the tail regeneration. So in the case of limb, we found that cells have to de-differentiate and acquire embryonic identity. In the case of tail, there are pre-existing cells, and that's how they complete regeneration. Now, if I have 5 minutes, I would go into the metamorphosis, which is the core theme of this conference. So for a long time, we have been interested in thyroid hormone and metamorphosis. Yesterday, you already heard from Jim Kaufman and Tom Lozito, how the metamorphosis are critical. And what is known about thyroid pathway is that whenever thyroid hormone, this is produced from the thyroid gland here near your neck, whenever it is produced, it drives cellular differentiation. It doesn't allow progenitor to stay in progenitor state, but it kind of pushes them into differentiated cell lineage. So you have, I was just mentioning, you have this PAC7 positive progenitor. They also kind of undergo differentiation and form muscle fibers when thyroid pathway is activated. When you have a neural progenitors, this neural progenitor go on and make neurons in the spinal cord and brain. Now, metamorphosis in axolotl is a very drastic and dramatic phenomenon. You can see that this is a neotenic axolotl, and the moment we give them a thyroid hormone, they lose their gills. You can see here. They lose their fin. The tail becomes much more rounded. Skin becomes very firm, and body kind of shrinks and becomes very tight. Now, logically, when you think about this, is very natural because animal is now going to move from aquatic environment to the terrestrial environment. And when you're walking on the land, there is a lot of abrasion, you suffer a lot of injury. And if you have this kind of a water pouch, then it can rupture and it will damage. So you need a firm skin. And that's why this is what happens during upon thyroid treatment. What is known about thyroid hormone is that it's a negative influencer on tissue regeneration capability. Probably Yunbo Shi, after my talk, he would be talking. He works with the thyroid hormone and metamorphosis in frog. And in the frog, when the metamorphosis happens, they lose their tissue regeneration ability. So before metamorphosis, they can fully regenerate their limb, but after metamorphosis, they cannot. And they only form this kind of a spike-like structure. Axolotl also, upon metamorphosis, loses this kind of a fully pattern formation ability, and it starts developing this kind of a defect. So this is a normally forming limb, this is a normal regenerated limb, and this is the defective limb. As you can see, the skeletal elements are less. So very briefly, let's look at the thyroid pathway. The thyroid pathway begins with this molecule called T4, which is circulating in our blood. It enters through this kind of a protein which imports T4 from blood inside the cell, this thyroid transporter. And then there is this molecule called DIO3. DIO essentially stands for deiodinase. It removes an iodine from T4. So DIO1 and DIO2 produces T4 to T3, and T3 is actually an active compound which goes inside the nucleus and drives the expression of certain genes. So when you have T3, certain genes are active. When you don't have T3, all these genes are kind of suppressed and they do not express. But DIO3 kind of degrades T4. So it also removes iodine, but it forms a reverse T3. This is an inactive compound. It cannot do this job. Okay, I'll come back to this later. But I want to highlight the changes that is happening in the skin here. So when the metamorphosis happens, this is your epidermis and this is your dermis. And you can very nicely see that dermis becomes very compact and it's rich of collagens. So what is happening here is that tissue kind of shrinks, and that's what we have quantified. We can see that epidermis reduces its thickness. Dermis also reduces its thickness. And we have also quantified cell density. Cell density kind of decreases. And then we have compared the proteins that are expressed in neotenic skin and metamorphic skin. And we find that there are enzymes which are responsible for degrading this collagen and ECM are downregulated, whereas there are some other proteins which are responsible for kind of blocking the function of this proteinases. They are upregulated. So this is how essentially the ECM, the extracellular matrix remodeling, is happening by not allowing it to degrade and allowing it to accumulate. And you can see in this slide, like, you know, upon metamorphosis, collagen goes up, fibronectin goes up. These are the extracellular matrix proteins. So in the last 1 minute, I want to highlight this DIO3. This DIO3 gene kind of upon amputation goes up and stays up for a very long time. And so what is happening here is that the fibroblast cells that are forming the limb blastema, the structure that give rise to the new limb, they have high expression of DIO3. This is a colocalization of DIO3 with fibroblast marker. And what we have uncovered is that not just DIO3, but the entire thyroid pathway is shut down in this fibroblast during axolotl limb regeneration, as if the fibroblasts are afraid of thyroid pathway, and they are trying to protect themselves from the thyroid. And this is how the blastema formation happens. So I'll skip this slide and go to the last slide. And that is this. So I think the arts still exist, but just in a different form. In the modern times, people no longer paint with these brush strokes, but there are competitions held for, you know, the beautiful microscope images. And these are just 2 images from my lab, which were done by my postdoc, Marco. And he won this 2 images. He won this 2 contest, basically. One is this axolotl soup bioimaging North America contest in last year. And then this year, axolotl in this Nikon Small World. So what I'm trying to say is that arts are still important and very relevant to the science. And with that, I would like to thank the people that work with me. This is my team here at MDI Biolab. These are the animal core. They are absolutely essential for our work that we do here at Biolab. And then thank you very much.
Thank you very much for a wonderful, wonderful talk. We will have lots of questions. Our neurons at the moment are going 160 or something like this, after Benin, after all these wonderful things. And then you finished with these things. I think Linda will have to respond. No more paintbrushes. Well, we'll see. You're first.
Could you just go back to the third slide before this? Yeah, that one, the summary. Yeah.
Yes,
Because I had a couple of factual questions.
sure.
So in terms of cell mechanisms of regeneration, so the axolotl is both, right? Pre-existing and differentiated. Is that what you found?
This slide, I can go back. Yeah, here. So
You went past that one really quickly.
yeah, previously I didn't go into the detail of this, but we have shown that when the limb regeneration happens, you have a differentiated fibroblast, and this fibroblast kind of go back to their embryonic identity, become de-differentiated, and this process of going back is called de-differentiation in scientific language. So they de-differentiate, become progenitor, and then these progenitors have the capability to give rise to fibroblast, skeleton, tendon in the limb. But in the case of tail, we find that this Meox positive population, they're pre-existing, they're sitting there, and the moment you have an injury or you activate them, they're capable of forming all this lineage that we discussed, like myogenic, skeletogenic, and fibroblastic.
And then my second question is in terms of the metamorphosis, this is like such a basic question, but do the axolotl actually metamorphose in their lifetime naturally?
So axolotl normally do not metamorphose. Axolotl is a neotenic animal,
Okay.
but occasionally, when there is a high iodine content in the environment, they are capable of undergoing metamorphosis.
Huh.
So very, very rarely this happens, but it can happen in nature. In the laboratory, all we have to do is inject some thyroxine hormone and they will metamorphose.
And so the thyroid pathway being shut down in the regeneration process, can you just tell us what relevance that has? Basically you're working on it for humans, right?
That's
so
right.
What does that make you think, given those, what do you conclude?
This is a very, very important and interesting question, and we have discussed this very long time. We should patent it, Herman. But I was more comfortable with this audience, so I didn't go into the detail of it. But we do think that by shutting down thyroid hormone, we could improve probably regeneration or wound healing.
We still have things like that. When we shut down thyroid hormone, you get a disease which is called myxedema, and your skin becomes different. And you all of a sudden, in front of your shins, you develop this watery skin you have seen in the axolotl. So it's all, in all the textbooks in medicine, and you use it for diagnosis, but we don't understand how this happens. Only when you look at this, all of a sudden, you get more of an understanding what myxedema means. And Prayag and I, we have MD PhDs working on that, and this is fascinating.
And iodine is important because thyroid hormone is essentially an iodinated amino acid called tyrosine. So when tyrosine amino acid gets iodinated, that's what is a thyroid hormone. Yeah, and this T4 refers to 4 iodine molecule on tyrosine, and T3 refers to 3 iodine molecule on tyrosine. Ondine, please.
Just beautiful talk, thank you. I was curious, you discovered basically a population of progenitors that can give rise to muscle cells. But when the tail regenerates, there's a lot of different cell types. There's endothelial cells and neurons and skin. And so who's giving rise to those, if not your collagen A1A3 population? Do you think that the heterogeneity of the stroma, there's one committed progenitor for each downstream cell type that's going to emerge in that tail?
That's right, and that's what is happening. So endothelial cells give rise to endothelial cells, nervous system give rise to nervous system, epidermis give rise to epidermis. But I have always been fascinated by this connective tissue lineage, the so-called connective tissue, because this mesodermal, mesenchymal connective tissue, whatever way you want to call it, this is the population that carries the identity of the position and also what kind of pattern needs to be formed. This information is residing in this particular cell type, and that's why we have been focusing on these cells.
Two of them are discussing is really very fascinating also for all the others here. Because for a long time, we are all concentrating in research on the structures which we can more easily identify under the microscope. So muscle or vasculature or bone. But these two are talking about all the cells and this messy thing which is in between, which is amorphous, which doesn't count really. And now we are finding out it's more important than all the other structures. And this was this discussion that from this amorphous cells sitting in there, doing actually nothing, that they are the origin of life.
So we have Youngbo.
Yunbo, yeah.
Hand raised. Youngbo, can you unmute and ask your question?
Yeah, very interesting talk. Very nice. I have a couple of questions, maybe very simple. The first is actually, you mentioned that in the tail, there are two types of progenitor cells migrate into the regenerating area.
Yep.
My question is, how do you rule out the possibility that maybe some cells, differential cells, migrate like a dermis to cover the wound? After migration, they de-differentiate into those two types of cells because you are using a constitutive promoter, not an inducible Cree. Is it possible that the de-differentiation now they express those markers mimicking the migration?
So Yunbo, I for the sake of the audience, I didn't dwell into the details, but these are all inducible Cree lines.
I see.
So this is indelible labeling. You cannot change the fate by changing the colors.
Okay, sorry, I didn't catch that.
Yeah, and maybe in the interest of time, you can ask a few more questions, but I'll arrange a separate meeting with you so that we can discuss the scientific details of it. Okay, because there are. I hope you understand. Thank you.
Sure.
One more question from you. Go ahead.
Okay, so the second question I have is on the limb regeneration, the thyroid hormone signaling pathway decrease. That's very nice. My question is, when you analyze those pathway, were you using tissue of the regenerated part, or you're using a mixture of both?
So once again, I did not go into the details for the sake of audience, but this is a FACS-sorted connective tissue population from PRRX1 positive inducible Cree line, which essentially labels only connective tissue cells. And then FACS-sorted cells were analyzed using both microarray and single cell transcriptomics. And that showed very clearly that it's the fibroblast-derived cells that they are scared of thyroid hormone.
I see. But my question is, did you isolate the whole limb or only the regenerated portion?
So we have a whole time course of mature and regeneration time points, and all these time points were FACS sorted for connective tissue cells. And that's how these experiments were performed.
Okay, thank you.
I'll set up another meeting with you because we have a lot to discuss. I know that. Thank you, Yunbo.
Thank you.
I have one more question, and that is, it seems to me that in all of this, a kind of central paradigm is that ontogeny recapitulates phylogeny. And that, we are seeing here in this metamorphosis from, an earlier evolutionary creature to, something that is higher up on the, later in the evolutionary chain, and that humans will, at the kind of branch of evolution, is, that will give us clues. Is there anything wrong with that paradigm,
Thyroid pathway. See, I'll tell you how science works. The science works with questions that you can answer.
Yeah.
And in order to answer a question, you need to have the right tools. If you don't have the tool, then we keep pushing those questions and we say, we'll study later or it's not important. And that's what happened to fibroblasts, because fibroblasts are cell type that is sprinkled across your entire body. And it's very, very difficult to study something that is not structured, like muscles are structured, skeleton is structured. So it's easy to study those tissues. But a cell type that is just sprinkled like a salt and pepper across body, for a long time, people ignored it because they had no tools how to study them. But now, in the last 10 years, we have put a lot of effort to develop specific tools that allow us to only label fibroblast cells. So then we can separate them from rest of the population, understand what kind of gene signatures they have, etc. So that's the key. So the same goes for the thyroid pathway and metamorphosis. What's happening in mammals is that thyroid gland organogenesis happens very, very early. Thyroid gland develops so early in mammals. And our neoteny is actually very short, like in the only first month in humans, basically. Then you are all metamorphosed. We are all metamorphosed already in mother's womb. So how are you going to study this question, which is there is no way to study this. You see the problem there. The same happens with mouse. Even before mouse pup is born, it is actually fully metamorphosed. So that's why people have not been studying it. And that's where axolotl and other amphibians can be of immense value, because you have a very separate neotenic development and very separate metamorphic identity. So now you can compare them and see what are the different things. Thank you.
I have a very different question. I can't help but reflect on your paper in light of Risham's talk and talking about her emphasis on the Nikisi-Nkondi figures and the notions of secrecy and access. And in that case, you had power figures that were invested with power. In this case, there's a recognition that there's a natural, an animal in the natural world that has a power invested in it already. And I'm kind of curious to hear you reflect a little bit on the role of the scientist in the interaction with this animal, and maybe thinking through, with the Nikisi-Nkondi figures, not only was the power hidden away inside the figure itself, but they were hidden away from kind of, let's say, broader public access. And I just, using that as a catalyst, I'm wondering if you could reflect a little bit on your role.
Yeah, sure. So the axolotl, as I mentioned, it comes from Mexican culture. It's found in Xochimilco lakes, which are almost now extinct because the Mexico City is built on that lake. Okay, but back then the Aztec culture, the indigenous people, Aztec culture, they have worshipped axolotl for very, very long time. Actually, the name axolotl, ajolotl, actually comes from the demigod that the Aztec people used to worship. So there is a long history of axolotl with the mythology and history, how the naming and everything evolved. They also had a lot of these concepts like, like if you apply axolotl's blastema to your wounds will heal better. And a lot of this mythological or superstitious, whatever you want to call them, beliefs originated, but there is a fundamental basis that they could see that axolotl can regenerate their legs and tail. And that is where these ideas or notions would have come, that if you apply, a blastema onto the human wound, probably human wound will heal better. Now, it's not an easy thing to do. The animal ethics committee would kill us if we go with this kind of an idea. But what I'm trying to say is that although on one hand, we feel the secrecy and all these things that are hidden to the knowledge, they have been there in the culture for a very, very long time.
But you were also asking about communication outside and secrecy and kept behind doors. You have realized that we talk about communication to the public all the time, but what Prayag actually did during the discussion, he talked to Yin Bo, and he actually said, let's talk about this behind closed doors. He used the argument because the audience and all that, but he was actually, what he meant is we shouldn't talk about this in public. We close the door because this is so powerful, and the spirits we're talking about are so powerful. We only can do this in private. I'm serious about that. This is, the language they use, you can't communicate that to a larger audience. It has to be small. And although we don't do this anymore, that we keep them out and we say it's so powerful, you can't deal with that. But in a way, it's still there.
There's one question at the back.
Oh,
first.
yeah, if someone else should ask a question.
Yeah, I just sort of riffing off that and something that you said earlier about tools strikes me as a way to come back around to the prompt that Rebecca had given us about requesting, especially our scientific friends and collaborators, to historicize the sort of state of the problem or the genealogy of the inquiry. And it seems to me that just in comparison with Risham's talk this morning, we could do it on the level of techniques and the advent, the derivation, the history of the tools being marshaled to pose, to frame, to articulate the questions in the first place. I'm thinking of Risham's appeal to X-radiography techniques and Pamela's proposition about what would art history look like if it had been modeled around these kind of artifacts, and whether or not X-radiography would be an appropriate technique. But we certainly, in our discipline of art history, maybe are increasingly cognizant of the ways in which technologies have been imported from other domains that have nothing to do with the study of art, but are very useful for asking certain types of questions. And then what happens once those techniques come to be institutionalized and turned into knowledge producing?
Well, yeah, absolutely. I mean, I get you. The whole field of science heavily depends upon technological progress, that not just biology, but every other field we are talking, whether it's chemistry, physics, astronomy, we have adapted methods from different field, like how, just to give an example, like how the way Hubble telescope works and how the light gets distorted when they are reaching to the human eyes on the Earth because of atmosphere. A simple thing like that has been adapted into the microscopy field, because we know that light gets distorted when they pass through different sample. So we have a separate project on those things as well. But I completely get you. I mean, technology is very powerful. We are asking the same question that 200 or 300 years ago scientists were asking. For example, the very first documentation of axolotl limb regeneration was done by this gentleman, Italian gentleman, Spallanzani. That was, I think, 1768. So he asked the same question: how do axolotl regenerate the limb, and why can't we? And if you look at the central question of my lab, it's the same. How can they, and why can't we? There's nothing has changed. The only thing that has changed in between this 250 years is the methodology. What was accessible before and what is now available.
I would get one not step further, but the technology determines actually what's happening. You just use the words like we are asking the same question, and then science depends on technology. Yes, but it also shapes and makes the science. So we are actually, the pictures you see, and you have seen these wonderful pictures, this is new technology answering old questions, but generating a whole new understanding how the
A
whole
huge wealth of
subject is
knowledge is
composed.
generated.
We generate our own new uh, uh, subjects using new tools.
And I mean, specifically on that, can you say for the humanists in the room, like where does the single cell sequencing technologies that you're using, like, can you give us a history of that? Like, where does that come from? Is it?
I could, but that would take a very long time. In brief, basically, a methodology was developed in 2008, which allowed a large-scale sequencing of any sample. And this methodology was then further adapted to single-cell sequencing, and then it went on to another methodology called spatial sequencing that a lot of labs are using. I'm happy to discuss this outside of this domain.
But it's really what I just said, it's generating new educational systems. We have to train all the scientists
Yes.
now, the young, using these methods. And now for the first time, critical voices. What you're doing here is you're centering on that. You're generating a field. You're not using it as a tool anymore, but all of a sudden we think along these lines. So it's a really very fascinating subject of tools and novel development. And as Prayag has mentioned, 2008, it's only
Illumina was
14 years, but at the moment it's dominating. It's dominating our institution on Dean. They're doing single cell backwards and forwards and in different areas. So it's very, very interesting. Okay, I think this
should
was
move on.
wonderful. Thank you very much
Thank
for the
you.
great discussion. And